Hello everyone, hello, can you hear me today? I hope you're all doing well. I'm so glad you could join me for the stream. I know it's been a while since I did a stream. Just been very busy, but I hope you're all doing well today, and thanks for joining me at the bench. And we're going to be doing some fun stuff today, basically. A lot of plasmid work. We've got our two mascots sitting here. We've got Bert and Ernie, our double-stranded DNA, and Eli Ecoli, of course. Here, let me switch up our soundtrack for a little bit here. Second. Let's put it up on our laid-back music. There we go. There. Let me know if the music is a little too loud for you and I can turn it down. Loud in my ears, but that's just my headphones. But yes, I'm so glad I could do a stream this week with everyone. So, as you saw from the title of our stream today, I'm going to be doing some plasmid work. So basically doing my very first streams that I did before, going back, and we're going to be making some plasmids, doing two different assembly methods today, and then one of those requires a transformation into Eli Ecoli right after it, so we're going to be doing a transformation, and I think I'll also do some plasmid prep, so some plasmid extractions, some plasmid DNA extractions today. So we'll get to hang out for about 2.5 hours. I hope that'll be awesome and that we can do some science together. But yes, welcome. Welcome. Thank you for joining me. Yes, okay. Alright. So I hope it hasn't been too long, and as always, if you wanted to catch up on any of my streams from the past, including my previous plasmid work, I have it up on my YouTube channel, and there it is for you. But yeah, so if anybody has any questions about the stream, let me know, obviously, of course, and I can explain what we're doing today. And if that doesn't work, then I can always try again. But yeah, so today I'm just going to be doing some more plasmid ligation work, since one of the plasmids that we did on the very first stream did not work. When we did it, didn't, like, any of the colonies didn't grow up. So we're going to redo that. I've kind of revamped it. Basically, I just changed up how much of vector to insert we're going to be doing. So vector is the backbone of the DNA that we're going to be using. It has the antibiotic resistance, so you need a gene that allows it to grow up on a plate that has an antibiotic. And the insert is the gene that we're trying to put into the backbone. And I think I'll try to do some drawings today in order to help explain that. We'll see how that goes. It's always fun. It's always fun when I try to do that. But yeah, so we're going to be doing that, and then we're going to be doing what is called Gibson assembly. And I will definitely explain that with a drawing because it's a lot easier to see it when it's written out. But it basically takes advantage of the insert and the vector have matching ends, and using 3 3 different proteins for it. It chews back the ends, they anneal. I'm just going over a brief overview of how it works. Then a polymerase will fill in those ends, so it basically looks like that, and then the ligase will put them together. But don't worry about it. I know it's a lot of jargony stuff I just said. I will write it out. It's a lot easier to see it on paper. So we're going to be doing that. So just a regular ligation and a hi-fi or a Gibson today. So that is the plan. That is the plan. So we get to utilize both of our mascots today. Yay! Although, if you've seen the transformation one, we're not going to be nice to Eli today. Unfortunately, poor Eli's going to get a little hot today. But yeah, so should I jump right in? If anyone has any questions or anything, let me know, of course. But let me move these guys over. And there, maybe. There. Make it a little easier for you to see. And I'm going to bring over my notebook and draw it out. So basically with my first stream, I don't know if I explained it very well, how we're going to be doing the ligation and everything. But today we have what I'm going to be calling a vector. So here's our vector. And then, what color should we do for the gene? Green? Does green show up well? Yeah. I think green's showing up very well. And this is a gene. Can you see that? Can you see that? You can always write bigger too, if you can't see that. Or I can use a different color. It's not showing up very well. Let's see. Can I use a darker color? Let's try the purple. Purple or better. Kind of. Yeah, a little darker. Okay. You can see the difference. I just want you to be able to see the difference, though. You can see the difference between the blue and the purple. Let's see. How about red? Oh, the red looks better. All right, we're gonna do blue and red. I guess we're gonna be a little USA patriotic today. So, when I say vector, this is gonna be our background that's gonna have AMP. So this AMP is short for ampicillin, which is an antibiotic. And this is a chemical that we put on the plate for Eli Ecoli to grow on. And if it has this vector, which we're going to put into Eli, it will grow. If it doesn't, it won't grow. So when I talk about the vector, basically has this AMP gene on here. So that's the vector. If you guys want to know the difference, in the gene is whatever we want to put into this vector, whatever we want to put into Eli for downstream experiments. So that's what we're working with today. And either ligation method that we're doing today, either plasmid construction method we're doing today, is basically just putting a gene into our backbone. So this is the background right here. Now, let's see, I'm just going to move that up a little bit. For a regular ligation, and then, there we go. For a regular ligation, we're just going to basically be putting these two together using one protein, and that's a ligase. So we'll have the vector right here, and our gene right here. And what ligase is going to do is it's going to work here and here where we have those open spots, and it's going to put them together. So ligase is going to work. Oops. Oh gosh. You know, I already can't spell today. It's fine. It's fine. Let me fix that. Let me fix my spelling. There we go. Make sure that's dry. The ligase works here and works here. And what it's going to do is it's going to put our two pieces together so it won't have those breaks. It's going to look just like this in the end. Now I know the DNA is going to look more like this, like an actual helix structure, but just for simplicity, I'm just drawing it this way so it's easier to see. So there won't be any breaks like you see right there. And so ligase uses the 5' ends that you see right here, there's going to be a phosphate on there, and it's going to use that to basically do a reaction to go boop, just like that on our double-stranded DNA. So that's a regular ligation that we're doing today. Now the other one is called, do I have enough room? Here, I'm going to switch it over to this side. Sorry guys, oh, we got to move you over. I need a little more space. A little more space. So this is a regular ligation, but we're also going to be doing what is called Gibson or hi-fi. So this is the other one that we're doing today. And this has, one more, I need one more color. One more color. Good color. Oh, here's, we'll just do black. Black always looks good, right? We're going to be using the ligase in here, as well as a polymerase. So polymerase is the enzyme that adds base pairs to a DNA strand. And then it also uses an exonuclease. And what an exonuclease does is it basically chews on an end of a piece of DNA. So it takes DNA base pairs off of a strand of DNA. So what's cool about hi-fi is it takes place, so we have the vector in our gene. So this is our gene. And I'm drawing it as a single strand, but remember these are our helix, our double-stranded helix. And here's our vector. So what's interesting is it uses, I need another, another good, we'll do green. Let's see green. This thing called homology. And that basically says that we have this short little piece on both our vector and our gene that match. So these little parts right here match to each other. And we added this by doing a polymerase chain reaction and using primers, which are small pieces of DNA that we order from a company. And basically, when we do a PCR, it adds what's on the primer to our gene. So we made like an oligo piece of DNA that uses the polymerase and puts it on there. So both our vector and our gene have this short piece right here that match. They completely match. So what's interesting is when you're doing the reaction, so here it is a little bit bigger. These guys, we'll actually make it double-stranded just so you guys can see it a little better. The exonuclease is going to come in, and we're just going to call it so, just to make it better. And it's going to chew up the 3' ends on these guys. So it's going to actually chew back both of these sides. So in the end, what you'll see is something that looks like this. Oh, wrong way. 3' just. 3'. I like to yell at myself a lot. So it's all good. I'm thinking it right. I always have to remember this. Because it's different. Yeah, other way. Let's change it up. It's actually, let me put these down. Too many things going on at once. Yeah. What it actually does here, because DNA is 5', 3', is that way. So let's do this. It's actually going to go differently down here. So the exo goes there. There we go. Let's make this longer. And then here is our right there. And actually choose back a little further than just the spot where it matches. So it's going to look like that. And then it's going to look like this on the other side, where our gene is. But choose back just like that. So we have these big breaks between it. So this is after the exonuclease. So what happens is, because these match, these two pieces match, and I'll make it a little longer just so it makes it actually match. They actually anneal, so you cause like a double-stranded product. So these are actually, I'll add a plus. They come together like that. And when it does, polymerase comes in and is going to fill in these spots. So then it basically, we're going to have polymerase come in. Polymerase is going to come in and fill in these spots. Excuse me. Sorry, I'm getting over a cold, so you'll have to excuse me sometimes. So it's going to come in and it's going to basically add where those spots are. Oops, no, wrong way. Going to add here. And then there. There. So it's going to add in those spots where it was not where it was, there was space. There. So now we have these nicks right here. So what happens is we need to fill in those nicks, so these spaces right here, because we can't have DNA broken like that. It's gonna get destroyed. We don't want that. So ligase comes in and does what it does for the regular ligation and puts those two together. So in the end, we basically have our plasmid put together with our gene in it, because before we didn't have this happen. So now it becomes a completely nice piece of DNA. No nicks, nothing. It went from being a vector and a gene, two separate entities, and using both the exonuclease to chew back the ends, excuse me, and then polymerase comes in, fills in those ends, and then to seal up these little nicks caused by the polymerase filling in, the ligase comes in, puts those together, and then we have our vector and our gene put together, and it should be a nice, basically circular piece of DNA. So it's going to look like this in the end, versus two separate things in the end. Hopefully that made sense. I know it was a little bit of mess ups there, but hopefully that makes sense. So if you have any questions, let me know, obviously, in chat. But that's what we're going to be doing today. A regular ligation and a Gibson or hi-fi in this case. So yeah. So shall we get to it? Are there any questions in chat? Just wait a couple minutes. We go through here. So yeah, so if there's any questions, obviously let me know. And if you have questions later on, feel free to put it in chat as I go along. Not a problem. I will get to them and hopefully answer them to the best of my knowledge. If it's not a complete answer, I will try to get back to you on that. Or chat might know. Always a good thing. There's usually scientists in chat that help me out, which is great. But yes, if there's no questions, I'm going to get started, because we need about half an hour incubation for doing the Gibson right here. This ligation, we won't be able to do the transformation today. For that, this is going to be sitting at 16 degrees Celsius overnight, so you won't be seeing the transformation for this one. But after the hi-fi is done, it takes about half an hour at 50 degrees Celsius. Then we transform it directly into the cells right after. So we'll be doing that. And I think while we're doing the transformation, I will do some plasmid DNA extraction, just to keep it going. Just get some science heading out. But all right, no questions. I'm going to get started. Okay, let's move this out of the way. We're gonna have to say goodbye to our lovely mascots for right now. They can always show up a little bit later, but I don't want to get them dirty, so I'm gonna move them out of the way as well. All right, so first things first, always PPE. I've got my lovely safety glasses on, my number one PPE. Second, of course, is donning the lab coat. And if you've been here for previous streams, you know that I wear a purple lab coat. So I've got my purple lab coat going. Alright. And last but not least, of course, our gloves. Make sure safety first before we start doing any work at the bench. Alright. Ooh, my glasses fogged up because I took them off. There we go. Okay, so I'm going to go grab the components that we need out of the fridge. They're sitting there waiting nicely for me. And let's get these ligation and hi-fi started. So, I just, for my own personal sake, I've already labeled which was got my vector and insert, and then for both of them, so A, B, and C, D, we're going to be using small tubes today for doing these reactions, so PCR tubes. Let me grab some small tubes. There we go. Let's label them A and B, and B and D. All right. Now we're going to be doing small volumes, of course. So we're going to be using our P10 and our P1 for doing these today. So first things first, make sure I have the right one. So A and B are what we're going to be doing for the hi-fi. I'm going to get this started just because we have that 30-minute incubation. So that needs to get going first. And I'm going to go grab our mix for that. So we buy it from a company. So it comes with a master mix with our proteins that we need. So the ligase, the polymerase, and the exonuclease in the buffer as well. So keep it in our little cooler since it does have the proteins in there. So it's right here, ready to go. But first things first, let me add our water, since if I'm gonna mess up something, that's the easiest thing to fix, is how much water. So we need 1.1 microliters of water, and I figured out how much I need for both the vector and our insert and how much water yesterday, just to make it easy for stream today. So I'm going to add the water first. Oops. Caught a tip from my thing. Sometimes my coat likes to do that. Add it into the tube, down at the bottom. That's all set. There we go. And then we're going to add the inserts, just because I have the pipette. We need 0.8 microliters of our insert, which is right here. It's a very small volume, very small volume. And I'm going to add it right down to where the water was, just to make sure that we're not putting anything on the side. I'm going to be spinning it down anyway, but it's always good practice to pipette straight down into the liquid when you're working with it. And we need 3.1 microliters of vector, which is right here. All right. Put it right down in there. There we go. And then last but not least, we will add 5 microliters of the hi-fi master mix, which is right here. Oh, it's a little frosty. Just let it thaw a little bit. Yay! We're doing the two things that I always do at the bench. Move colorless liquids from one place to another and wait for things to thaw. And then later we get to wait for our centrifuge to stop spinning. So we're gonna hit all the things today. Just gotta wait for it to thaw. There we go. Going. Mix it up at the same time. There we go. Alright, 5 microliters. And then right down There we go. And I'm going to just increase, just by a little bit, the volume so I can mix with this. So I'm not going to vortex it because there's ligase in here and ligase does not like to be vortexed. So I'm just pipetting up and down to get it thoroughly mixed. There we go. And then what I'm going to do next is I'm going to briefly spin it down just to make sure that if I got like a small droplet on the side of the tube or anything like that, it'll come down. I'm not going to mix it enough so that all the components just sink to the bottom. We don't want that. But I just don't want something really important sticking to the side of the tube, and that kills the reaction. We don't want that to happen. And then I'm going to put this at 50 degrees Celsius in our PCR machine, which unfortunately you can't see just because the camera is fixed, for half an hour and get that going. So, need to change. All right, that's all set. Let me move on to our next one. So we're all done with this one. We're going to do CND next, which is just our regular ligation. So all it takes is the ligase and the buffer. Let me go grab the buffer from the freezer. Since I have a little, hopefully, a little aliquot of it. Do I have, I don't. All right, just a second. All right, there we go. Sorry for that. I need to grab another small tube of our ligase buffer. So that's basically the salts and the substrate that ligase requires. So ligase requires ATP in order for it to actually work and do the mix. So that's already within the master mix that we did for the high-five. But we have a separate one for doing just a regular ligation. All right, so here's C&D. So first things first, of course, water. We need 2.1 microliters of water. And there we go. There. All right. And next we need, so for the vector, I need 4.8 microliters. So remember, the vector is the backbone, which has our antibiotic resistance gene on it. So it'll allow it to grow when we add antibiotic to the plate that we're working with. And you'll get to see the plate later if you're able to stay with me for the entire stream today. I will show you that. All right. Right down in there. Done. And then, I'm going to keep that right here. We need 1.1 microliters of our insert. So our insert has the gene that we're trying to put in here. Right down there. Perfect. Now I need 1 microliter of our buffer, which is right here. That's why it's so, to show why we have it in these small little tubes, the ATP that I was talking about, so the substrate that ligase requires, if you freeze thaw it too much, which means you take it out of the freezer, let it thaw, then put it back in the freezer, let it thaw, it actually starts degrading it so it no longer works. So we always aliquot it into small amounts so we don't have to worry about an entire tube of the buffer going bad just because we keep bringing it out of the freezer and taking it out again. All right, so that's all set. So now we add a microliter of our ligase. So this is a protein that's going to put two pieces together, our vector and our insert. Okay. That is all set. I'm just going to put this T4 so I can use it for later. Now, similar to what I did before with the hi-fi, I'm just going to change the volume up a little bit so I can do a mix. Since ligase does not like to be vortex, so you have to be gentle with ligase. I'm just mixing the volume up and down a couple times, make sure everything's all mixed. And then I'm going to spin it down, make sure anything that was sitting on the side that could have gotten there isn't there anymore. We'll do that real quick. Those should be all set. And then this one is going to sit overnight at 16 degrees Celsius. So I'm going to get that set up. I will be right back. Alright, I'm back. So, the last thing to do, obviously, is to put stuff back into the freezer, since I'm all done with this, and we don't want our proteins to get degraded just because it's sitting here. This should keep it cold, but I'm going to do that, and give me just another minute. All right, I'm back. Okay, so that is all set. I'm done for right now, so I'm going to take off my gloves, wash my hands, just in case. Always good practice at the bench. Get that all done. All right, so while we're waiting for the hi-fi to finish, which will be less than 30 minutes now. Let me just pull it up so I see it in our machine so I have a better idea of what's going on. The next step would be to transform it. And if you've seen the previous stream or saw my VOD about it, basically what we do is we just be really mean to Eli Ecoli and bring back our guys. We have Eli Ecoli. Here he is, our cute little mascot. What we're going to do is we're going to essentially force the plasmids that we are making at the hi-fi into Eli Ecoli. Now to do that, we're doing heat shock. Basically, we're going to heat up Eli Ecoli so that little pores form in his membrane, and the plasmid DNA is able to get into it through those pores. And then we'll quickly put him on ice, stop that heat reaction because we don't want to, kill poor Eli. We don't want that to happen. And then we grow him up and make him happy again, so that hopefully, if the plasmid's there, then he will survive when we put him on antibiotics. So that's just the transformation in like very quick terms. So if there's any questions, let me know. I know this is different from what I did last time, which was sequencing, but that's okay. Hopefully I'll be able to move on to sequencing soon. Just waiting on samples. We're just waiting on samples. But I hadn't streamed in a while. I wanted to do a stream again, and I always have to do cloning or plasmid work. So I figured, why not? Why not do another plasmid stream with you guys? And I get to talk about a different way of constructing a plasmid. But yeah, so what I need to do to get ready for the actual transformation when we're set is grab my ice bucket. So when it gets closer, I can go grab some ice. We're gonna need ice for the transformation, so here's the ice bucket. Then we're gonna move these guys back. So that'll be all ready to go. When we're set, I'm going to set our timers. Oops. No, stop. I don't want you to go. So there's an initial incubation on ice. Then our heat shock is a minute. Then to stop the heat shock, it's 2 minutes on ice. And then we grow them up for an hour at 37 to get them all happy again. So the initial 30-minute incubation is to allow the plasmid DNA to basically stick onto Eli Ecoli's membrane. So basically, while he's sitting on ice, plasmid DNA is going to come in and go sit right there, plop, right onto him, right onto the membrane. And that's so that when we do the 1 minute at 42 degrees Celsius, when we make those little pores in Eli's membrane, plasma that's already sticking there can just go right inside of Eli Ecoli. Then, after the minute at 42, then we stick in 2 minutes on ice. That'll stop the heat reaction, keep Eli from dying on us. And at 37, they'll grow nice. They'll be happier at that time. Basically do a spa treatment, pamper them before we decide to put them on a plate with the antibiotic, which the E. coli that doesn't get the plasmid, unfortunately, will not survive. But if they do get the plasmid, then they will survive and they will show as little colonies, like little white dots on the plate. And we've seen that before in my previous streams. So I don't think I'm going to be doing that just because this is the only time that I think I have this week in order to do a stream. That's okay if you want to see what it looks like. Excuse me, I have to cough. Sorry about that. Like I said, I'm getting over a cold right now. But yeah, if you want to see what the plates look like with colonies after growing it up, you can go to my YouTube channel where I have posted all of my previous streams. And there you go, I put the link in chat for you, and you can see what it looks like. And then during the transformation, while we're waiting, so we have the 30 minutes, then the quick heat shock and recovery, then the 1 hour, we can do a plasmid prep or plasmid DNA extraction, which I've done also before on stream, just because, you know, I've done it before, I've shown it, so it'll be easy to do, fill up that time, and I need to do it anyway. So why not stream it and spend some more time with all of you at the bench today? Oops, I need to move my things around. A little more space for myself. There we go. But yeah, so are there any questions in chat for what we're doing today? Once again, thank you for joining me at the bench. Always nice to see everyone, and hopefully you're enjoying the music, hopefully you're enjoying the vibes today. It's the middle of the week, so hopefully it's been a good week for you guys as well. Alright. No questions, that's fine. That is perfectly fine. Yes, alright, so getting ready for the transformation. I think I'll give it another, how much? 20 minutes. So about 10 minutes, I will go and grab some ice so you guys can chill out for a little bit, just because the ice that I have to go get is down the hall. It's a walk for me, so you can chill out with the music. But you know what we can do to make it a little easier for myself later is get my tubes and my columns all ready for the plasmid extractions that we're going to be doing. Get my prep set, since this is what I normally do while I'm waiting anyway. Alright, so we need our initial ones. So I got 8 to do today. So the plasmid preps that I would like to do today are from previous floating. We're just gonna see if it worked. They're kind of old, but that's all right. If it works, I'm gonna be happy. They grew, so that's awesome. I'm happy about that. Yeah, so we're gonna need that. And then let me grab the columns that we use. And then I just gotta label them all. And then we'll be all set and ready for the extraction when we start the transformation. There, perfect. Okay. So we can just chill while I label all of these. And of course, if you have any questions, you can pop them in chat. I can read chat while I'm doing this. It's not a problem. Alright, we're all done. Got them all labeled, so they're just gonna hang out until we're ready to use them. You can go right there. Perfect. I'm taking these off, but I don't want to use them on the computer, just in case. And I'm going to just briefly, I'm not gonna do a full, because we didn't actually do much, but I don't know, peace of mind to get my hands all clean. Just use some hand sanitizer. Alright. Oh my headphones keep slipping down. There we go. There we go. Much better. Oh, but yes, I hope everyone has a good week so far. It's been pretty nice around where I am. For those of you in the US, it's been a short week since we had Labor Day on Monday. So this is really my Tuesday, even though it's a Wednesday. But that's alright. It was a nice holiday weekend. We had some good weather where I'm at. Hopefully you guys also had some good weather where you were at as well. Got to enjoy it. And then, let's see, and I'm back at the bench doing the stuff. But yeah, hopefully, like I've said before on past streams, we're waiting for samples to come in, because what I really want to do is show off the sequencing that we do in our lab. So hopefully the samples will come in, and I'll be able to do some more streams. And it's going to be a lot of the same kind of thing, where I'm showing off our sequencing protocol, but that's fine with me. I get to spend time with everyone, get to talk science. For all the newcomers that come in, they get to see all the new stuff that we're doing at the bench. So yes, welcome to the new people that have signed on. Welcome to the bench. Today we're working on making some plasmid DNA. We just got that set up, and then we're going to be transforming that DNA into our E. coli, and that'll grow up. I'm not going to show the rest of that just because this is the only day that I have time to stream this week. But if you're interested to see the whole protocol, I do have that up on my YouTube channel, where I went through from basically what we did today, which is constructing the plasmid, transformation, growing it up from a plate, and then doing the plasmid DNA extraction. We are going to be doing a plasmid DNA extraction while we're doing the transformation today. So you get to see two of those. I just won't be able to show you what it looks like on a plate once it's done. So yes. Sorry, if all of a sudden my microphone goes off, it's just because I don't want to cough into my microphone and you guys don't want to hear it. I don't think you want to hear me cough. That doesn't sound good. You don't want to hear that. So you'll have to forgive me. You'll have to forgive me on that. But yes, I know it's been a while since I've done a stream, and I apologize for that. It's just been busy getting school started, so trying to get back in the school routine and everything, but I'm happy I was able to do a stream today and get back into the groove of things. But yeah, so any questions, if you have any science questions, it doesn't have to be about what we're doing today. It can be about just science in general. Go for it. Or want to chat about something, just put it in the chat. Or we can just hang out here and vibe with the music. I'm up for all of that. So I'm okay. And then hopefully I'm working on, I know if you hear pops and stuff for the audio, I apologize for that. It's something that unfortunately Mac is set up for it. Working on trying to get that audio fixed, so fingers crossed. Fingers crossed the fix that I'm working on will actually work and you don't have to hear that horrible staticky pops that happen all the time. So hopefully that'll be soon. Maybe this week. We'll see. I'm excited. But let's see, how much time do we have left on this? 12 minutes. Okay. So a couple more minutes and I'm going to go grab some ice to get our transformation set up. And then we'll get the transformation done after the 12 minutes. And we'll start working on the plasmid DNA extraction. And the DNA extraction is the exact same way that I did it before. So, not promise from a kit, we use different solutions to go through that, where we lyse it, which basically we break apart our E. coli to get the DNA out of the cells. Then we clean it up. That's basically the extraction protocol in a nutshell. But yes. Hopefully no one else is dealing with a cold. You know, going to school, the kids come home with colds. Even at the beginning, what can you do? Make it through, power through it. I'm on the mend, just every once in a while have a tickle in my throat. So that's just the problem that can happen. But yay! Okay. So we're getting closer and closer. Closer and closer. Okay. So I think it's about time. Yeah, alright. So I will be back. I'm going to go grab some ice, which is down the hallway, so it takes a couple minutes. And you guys can just hang out and enjoy the music. All right, I'm back. So, as you can see, I went and got ice. I also got some of our cells that we're going to need. So these are Eli Ecoli cells we're going to use to transform our hi-fi Gibson reaction into. So yes, hopefully that'll go well. We only have a couple more minutes and we'll be ready to see that. Let me double check. Yeah, so about 5 minutes, we'll be all set. So this is gonna just hang out right here until we're ready. Ooh, here, let's see. Here we go. Grab some gloves. I just want to be able to show you what the E. coli cells look like. So hopefully you can see they don't really have a color to them at all. But they're a little cloudy compared to water, right? Because here's water. So this is what E. coli looks like sitting in the tube getting ready to be transformed. Yeah, there we go. So, like I said, in past streams, we actually make our own E. coli cells for doing our transformations. And these are called chemically competent cells. Basically, it just means that we have them in a buffer that allows them to have the heat shock occur with them. And it doesn't completely wipe them out, doesn't completely kill them. And helps with the actual, pore maintenance in the membrane and allowing for the DNA to stick to the E. coli. So that's why it's called chemically competent. The other ones that are available are called electrocompetent cells. Those are in a different buffer because you use a machine called an electroporator that will use an electrical current to put pores into the membrane in your E. coli. So we don't have one of those. Those are expensive, and heat shock works just fine in our lab in that case. But I have done electroporation before. It's real quick, and it makes like a little zap sound when the current goes through the E. coli. So you know that it worked, but it could also make a sad zap sound. And if you hear that, that means that you did just basically kill all of the E. coli. So you don't want to hear the sad zap sound. And it's a very specific one, so I can't really explain it other than as a sad zap sound. You have to be able to hear it in order to know what I'm talking about. But yeah, so the only way that we can really mess up the heat shock is if we leave it at 42 degrees Celsius for too long, and that will kill our E. coli. And 1 minute, perfectly fine, we're good. And then we stick it on the ice in order to stop the reaction. So, not a problem. We don't have to worry about that. We don't have to worry about a sad zap sound in this case. But yeah, okay, so what are we going to need? I'm going to need my tips, get this ready. So we're going to transform the entire reaction, which is 10 microliters, into our cells right here. So hopefully we will get colonies with the hi-fi. I tried this before, and sadly it did not work. I got no colonies. So after some troubleshooting, hopefully this new reaction will work a lot better. We'll find out. And maybe next week I could do, if we get colonies, I can do the plasmid DNA extraction for that. We will see. We'll see. It always depends on what's going on as far as experiments go for me and as far as streaming. So that's why I don't unfortunately have a consistent schedule right now. What I really want is once we have the samples going for the sequencing, then I'll have a much more consistent schedule because that's all I'm going to be working with. So you can look forward to that. Hopefully soon. Let me check. That's a minute. All right. Then I will grab the tube out of the machine and we'll get this transformation started and see how it's going. Oh, I need to turn on our shaking incubator. So our shaking incubator basically is a machine that we use to... It's set at 37 degrees Celsius, which E. coli loves, but it also shakes at the same time. So when we're doing the recovery, which is basically the spa treatment for our E. coli after we do the heat shock, we want them to move in the tube because they don't like to just sit there at the bottom. They get really not happy if you do that. So it'll shake it at the 37 degrees that we have it set at. So that's why it's called a shaking incubator. All right. Perfect. Here we go. Okay, so here we go. Here's our reaction. It was set at 50 degrees Celsius for half an hour. So this is for doing the Gibson Hi-Fi. Just gonna let it cool down just a little bit. It's a little warm still. But don't need this anymore. If it cools down after a few seconds, we don't want it to be too hot going into the cells. All right. Yeah, there we go. What I'm going to do is I'm going to pipette the entire volume out of the tube. And then stick this on the tubes like this. So you can see that pipetting right into the, without making any bubbles. Seal it up. And then we tap it to mix. We don't want to vortex the cells. That will kill them. That would be bad. That would be bad. And now they sit on ice for 30 minutes. So within this 30 minutes, that plasmid DNA is going to stick to the membrane of the E. coli, and it'll be ready for the heat shock. All right, so while that is going, let's get going on our plasmid extractions. So, as I said in my previous stream, I just show you what I specifically do. And labs may do this differently on how you do the plasmid extractions. Let me grab these tips right here. Move this guy a little further so I have a little more space. Yeah, there we go. And I'll grab my tubes. So yeah, so I grew these up last night from a very old plate, but they grew, so I'm very excited that hopefully these will work, because this is a plasmid that I need to get for some later experiments. But we've got 8 to work with, and I'm going to set this to 750 microliters. And what I'm going to do is, they're sitting in the fridge, frosty. I'm going to pipette about 1.5 mils of our grown-up E. coli solution into these tubes. And what I do in these tubes is I spin them down in the centrifuge for a minute at 18,000 times G, so very fast, extremely fast, to pellet the cells, because the cells are sitting in this liquid. So let's see. Oh, I have one right here. So this is what the liquid media looks like when there's nothing in it. And this is what it looks like when something's grown. So you can see that it's cloudy. Hopefully you can see that it's cloudy. So this means that we actually have E. coli grown in here. So I need to be able to separate the cells that are in here from that liquid media, since that's what we're going to be extracting the DNA from, is the cells, not the media. So I just do this for all of them. I'm gonna try that again. There we go, much better. So I just redid that pipette hole because there's a big old bubble in the middle of it. And you know, honestly, it's okay that we're a little rough with these cells, because they're just going to be, unfortunately, laced and extracted. So we're gonna be even rougher on these cells. So get this done, and in the end, we will have some nice DNA. There we go. Okay. I'm looking for specific labels. Here, And yeah, I hope everyone's enjoying our nice chill vibes. Welcome to any new viewers that have just come in. Welcome to the bench. We're doing plasmid work today, so earlier we constructed some plasmids using 2 different protocols. And we just started our transformation protocol, which is putting one of those plasmids into E. coli. And now, while we're waiting, I'm doing an extraction for plasmid DNA. Because we've got time. Why not? And normally this is what I'd be doing in lab. I'm usually not just doing one protocol. If I have long incubation times like I do with transformation, I'm busy working on something else at the same time. So, get that multitasking skill up. Get that multitasking skill up. Alright, I gotta change some of these labels because some of I had initially picked 10, but some of them didn't grow up. And like I said, these are from some old cultures, so that doesn't surprise me. Some of them didn't grow up, which is fine. These ones did. I'm actually very surprised. But pleasantly surprised. And if these turn up positive, which I will do off stream, is doing a check, I will be extremely excited. Extremely excited. Because that means I have this plasmid ready to go, and I don't have to work with it anymore. Yay! Okay, so we've got all 8 of them in their tubes. They're ready to go. I'm going to go put these in the fridge since we don't need them anymore, and it'll give more space to work with on my bench. I'm going to grab the solutions that we need. So using various solutions for doing this. Might as well grab them all. Ooh, I need a waste beaker since we're going to be basically generating some waste. Yep. I need somewhere to dump the media out because I'm going to be separating the cells from the media right now. Okay, let's get this set to, I need 250. Excuse me. There. That's set and ready to go. Alright, so now I'm going to centrifuge these guys for a minute at basically 18,000 times G. Very fast. Just think of it as extremely fast. And what it'll do is it's going to pellet the E. coli cells that are in that media and will let me pour off the media so that we can just work with the cells which has our plasmid DNA within it. And then this is all set, grab that. But yeah, so I'm just showing you how I do this protocol. And if you're wondering what these paper towels are for, you'll see in a second. You know what? Here, let me move this over so it's more central to what we're looking at, since this is what we're actually doing right here. So it's easier for you to see since I was working off to the side. We don't need to see the ice bucket with the tube sitting in it. Don't need to see that. We know that the tube's sitting on ice. And this is almost done. But yeah, so the next step are using these different solutions, and what we're going to do is we're going to resuspend them in this solution right here. It's a buffer, just a resuspension buffer. This solution right here is our lysis buffer. So this is what's going to break open poor Eli Ecoli and spill out its cellular contents, including our plasmid DNA. And so this basically has some detergent in it that breaks apart the cellular membrane. And then we don't want it to do that for too long, because if you let it sit in the lysis solution too long, it actually starts to degrade the DNA. So in order to stop that, we use a neutralization solution, which is the other buffer, to stop it. All right, I'm just grabbing our tubes out of the centrifuge. There we go. All right, here's our tubes. I'll send this back. Now what I'm going to do hopefully you guys can see it. If you guys can see it, there's a pellet right here. This is our cells that I was talking about. So these are our E. coli cells that I just separated from the media. What I'm going to do is I'm going to pour off the media into our waste beaker. Stick it on the paper towel, upside down. Now I do this to get rid of as much media as possible. I want to have a clean pellet to work with. And the media contains, you know, all the stuff that E. coli likes to grow in. But it's not good for making a clean DNA prep. Because that can gum up what's on here, which is our column. We don't want that. We don't want that. So I get rid of as much media as possible. And that's why I use this. And then I just do tap tap. The tap is get rid of that media. Once I don't see it showing up on the paper towel anymore, we're all good. All we have left is our cell pellet at the bottom. So other labs might do it differently. This is how I do it. I like my little tap sounds. I don't know if anybody else in the lab likes it, because all they hear is tapping. But it works for me, and I get clean preps out of it, so I'm happy. I'm happy about it. And thankfully I don't do it all the time. I could see it getting pretty annoying if all you hear is tapping throughout the entire day. So if that were the case, I'd probably do it when nobody was here. If I had to do it that many times. Thankfully I do not. So it's only a short amount of time that you hear tapping from over here. But alright. Almost done. Almost done. There. Alright. Now, just because I like to keep my bench clean and have plenty of room to work with, spray that down with ethanol and wipe it up. Wipe it up. Get rid of anything that's sitting there. There. So it's nice and clean. There. Okay. So, now that we have just the cells in our tube, I'm going to add the resuspension buffer, which is right here. It's 250 microliters. Now, I'm not going to change tips because I'm not going actually down into the tubes. If I was going down into the tubes, I would definitely change tips. And if you feel more comfortable doing that, more power to you. This is just so I don't have to waste tips when I'm doing the same solution. I can just stay above the tube. And then what we do is we actually resuspend it. So I'm just going to head up and down and resuspend that pellet of cells. So that's actually in solution. So there we go. So now it's a nice clear solution. We don't see the pellet anymore. Yeah, perfect. And I'm just going to do that for all of them. So see how there's a pellet right there? You just basically break up that pellet, get back into solution. So you can't see the pellet. Now it gets that cloudy liquid. Cloudy solution. Alright. Now we want to resuspend it after because the lysis will go a lot smoother if you have the cells in a suspension versus as a pellet. Because the mixing, we don't mix the lysis using a pipette because then it could break up DNA too easily. So we mix it by inverting the tube, so basically taking the tube. Now if it were a pellet still, it would be extremely difficult for the lysis solution to get to all the cells. So that's why we resuspend it in solution instead of keeping it as a pellet. Alright. There, all of them have been resuspended. And put that away, we don't need this right now. So I'm going to add 250 microliters of our lysis solution. So this is where we're going to break open those E. coli cells and get all the DNA and whatever else is in the cells out of there. So remember, from biology, these cells are going to have DNA, proteins, lipids, all the things. And the lysis solution is just going to break all of that up so we can have just the DNA after cleaning. Okay, so I got to be quick with this. We don't want it to be in the lysis solution for too long because the detergents can start breaking down the DNA. And this is how we mix it. So what I was talking about, we invert the tubes. If they weren't in solution, those pellets aren't going to be moving. And that lysis solution is not going to do its job very well. All right, so you don't want this to sit longer than 2 minutes. After 2 minutes, that's when you start to see your DNA degrading. All right, so there we go. Nice and mixed. I'm going to set this to 350 microliters. Then we're going to add, this is our neutralization buffer. So this is going to stop that lysis before it starts degrading our DNA. And then same kind of thing, we're going to invert it to mix. All right, 2 more. And done. So this is going to stop that lysis real quick. Oops, oh gosh, I'm just chucking that stuff everywhere. We're just going to do this. There, done. So that has stopped the lysis. Now we don't have to worry about our DNA being degraded because of that. So next step is I'm going to centrifuge these for 10 minutes. The 10 minutes is basically because we want to spin down all the extracellular components, so those proteins, those lipids that we don't need that are now just floating in the solution with our DNA, and keep the DNA in a solution. So 10 minutes, that'll allow all of those cellular components to just basically become a white substance. I will show you. It looks like a white pellet when it's done. And everything that's in solution is just going to be our DNA that we are going to move forward with. So I'm going to set this, get this going. Oh. All right, and that's at the same speed that I spun it earlier, which is at about 18,000 times G. So while we're waiting for that, I'm going to just clean up a little bit, put these solutions back. We don't need these buffers right now. We are all done with them. And then I'm going to wipe up my bench just a little bit because I got some of the buffer on there. And it can get sticky. Oh, just a second. I need to get some more paper towels. I'll be right back. Right, all right. Had to go grab some more paper towels. I ran out of paper towels. Okay. This will help clean up my bench, and it won't be sticky from the buffers. Much better, much better. Okay. So, we still have basically 12 minutes on the 30-minute incubation for a transformation. So while we're waiting, here are our next buffers that we're going to be using. I'm just going to get everything ready. I'm going to need 800 microliters. So once the centrifuge is done, which is, like I said, one of the things that, as bench scientists, wait for a centrifuge to stop spinning, we're going to move the liquid from the tube into those blue columns. That's what the little blue things were. They're called columns. And it only fits 800 microliters. That's why I said at 800 microliters, which is fine. That's about how much we will get out of it. And what'll happen is the DNA is going to stick to that membrane in that column. So the column has a little membrane. The DNA is going to stick to that membrane, then we're going to clean it up using these buffers right here. So basically get rid of any extra salt that we don't need from the other buffers, because downstream applications, so if I want to sequence it or do something else, extra salts can actually inhibit some of the enzymes or the proteins that you work with in order to look at your DNA. So you want to clean it up really nice. And that's what these buffers do for us. So it's just a series of cleaning steps after this. But yeah, so any questions in chat? Anybody need a refresher as to what we're doing today? If you just joined us, welcome. Just working at the bench doing some various plasmid stuff today. But yes, if you have any questions, let me know. While we're waiting for centrifuge to stop spinning and our incubation on our transformation. We've got 10 minutes. 10 minutes left. So yeah, any questions, I'll be happy to take them. And also, if you would like to join our community outside of the streams, I do have a Discord server. I'm going to put that on there. And if you just joined us and you have no idea what I'm talking about and you wish that you had been here for the beginning of the stream, don't worry. I always put the VODs up on my YouTube channel, which I will put on here again. So you can always catch it later. And of course, Twitch has the VODs up for, I think it's now only 7 days because I'm not affiliate or partner. It'll only keep it for 7 days, but they will be there on my YouTube. And if you're enjoying what you're seeing and you would like to know when I go live again, just hit that follow button and the bell button for notifications, and it should let you know next time I go live. If you would like to follow me on Twitter, I also post when I go live. So you can follow me here, Scientist at the Bench, and you can get notifications from that as well. So yes, just my shameless plugs. My shameless plugs. So yeah, not a problem. The VODs that I have up on the channel are basically what we're doing today, except for growing up and what the plates look like after I put the transformation on the plate. So you won't be able to see that, but you can check it out, previous ones. You want to know what it looks like after the transformation is done. And then I have a sequencing protocol up there as well, because I did some sequencing. Then my big hope for the channel is that it's going to be sequencing. And then I'll have a consistent schedule of sequencing. And that'll be fun. Because we got the other channel working, our other computer working. OBS is finally working, so we'll be able to raid that channel, and you guys can see in real time the sequencing that we're going to be doing. So I'm very happy. I'm very excited about that. So excited about that. But yeah, so hope you're enjoying yourselves. Ooh, which reminds me, it looks like some of my caption, I'll have to make the window a little bit larger. Just because I do see it cutting off. Ooh, ooh, ooh. There we go. Forgive me while I... There. Hopefully that changes. Okay. It's just cutting off some of the end of the captions. And I want you to be able to see all of the caption if you're reading on it, which I'm sorry if it captures the wrong words that I'm saying. Sometimes the science doesn't go through. which can be funny, depending. But yeah, so let's see, we've got 6 minutes on that. Oh, the centrifuge will be done first. So I get to see some minutes on lunch. Oh, I'll be ready for lunch after this, definitely. I'm getting hungry. I'm getting hungry. I can't wait for lunch. But yes, so the centrifuge will be done before our transformation incubation, which is fine. What'll happen is do the centrifuge first. I might be able to get that spinning, and then we can go do the heat shock. All depends. You get to see like real time scientist multitasking. You can figure out where it's like, I can sit for a couple minutes there while I do something else. Make those decisions right at that time. But this is what I usually do. Welcome to the lab. Welcome to the bench. Always fun. But yes, here's our lovely transformation tube. He's still hanging out right there for another 6 minutes, 5.5 minutes, and we'll be all set and ready. And we'll move forward with that. We're actually going to be done the plasmid preps before the transformation's done, so that'll be nice. I can just hang out, and if people have questions, I can always answer the questions while we're finishing up the transformation. I'm okay with that, or we can just sit here and vibe with the music also as well, which not a problem. I just want to say thank you, lurkers. You're amazing. I love you. I'm a lurker myself. I'm just glad you're here, listening, watching the stream. You get to get some science in your day. It's always nice. Got a minute and a half on the stage. Yeah, so what did we do today? We did the 3 things. At the beginning and throughout this entire time, we're moving colorless liquids from one place to the other. The very beginning for the plasmids construction, we had to wait for something to thaw, so that's number 2. And now, the 3rd thing, wait for a centrifuge to stop spinning. So we're hitting all the scientist things. Always good when you can check off all 3. Oh, so close. We're so close. The 10-minute spin. The rest of the spins are only a minute, so they're much faster, since they're just cleanup steps. It's just back and forth, which isn't bad. This is the only long part of the protocol, essentially. And the transformation is, you just wait, lots of incubation times. There we go. So I get to hear it beep, unless I catch it. I might be able to catch it before it starts beeping. Oh, I caught it before it stopped beeping. Aha. Alright, so I'm just taking my tubes out of the centrifuge, and I will show you what they look like after the 10-minute spin. And set this back down a little bit. There. So don't actually do a 10-minute spin next time. Alright, so we did the lysis, and this is what it looks like now. So that white stuff right there, that white pellet, is all the cellular components, those proteins, the lipids, carbohydrates that are sitting in there, the big ones. So those macromolecules, that's what that white stuff is. And our nucleic acids, so the DNA, is going to be in solution. So what we're going to do is we're going to take that out very carefully and put it in these columns without getting the white stuff. Good if I have my labels. There we go. And I'm just going to get rid of this. We don't need it anymore. We don't need to keep those pieces of the cell. We got what we wanted, which is DNA. That's what I'm doing. Put it into this column. So this is the column right here. That white part right there is where the membrane is in the column, and that's where the DNA is going to stick. Just do this for all of them. Hope you guys can see what I'm doing. Closer to 6. Always good to remember where I'm sitting at. Pay attention. There's been many a time where I have put the wrong one in the column, but it's fine because you can just change it out. See these columns come out? So I can just renumber the column if I make a mistake. Because I have done that before. So it's okay. Alright, we're almost done. Oh, see? See what did I just say? I just put 4 and 3. Look at how easy that is. Done. Done. Yeah, see, I just wanted to show you exactly what the mistake is, so if you do it, you know what to do. You know what to do. Alright, there we go. Okay, so now we just centrifuge this for a minute, and we don't change the speed. The speed stays at 18,000 times G. And what's going to happen is the solution that the DNA is in is going to flow through, and the DNA is going to stick onto that membrane. Let's get it in centrifuge. Alright, so the centrifuge is going. Oh, and guess what? Perfect timing. Let's go do the heat shock. So I'm going to do the heat shock. So what I'm going to do for a minute, I'm going to put our tube that's been on the ice for half an hour. So this is our E. coli with the plasmid that we made to get 42 degrees Celsius for a minute. That's going to cause pores in the E. coli membrane. Let that plasmid go in, and then immediately afterwards, I'm going to put it on ice for 2 minutes. So let me go do this heat shock, and I will be right back. Alright. So, I finished up the heat shock. It's sitting on ice for 2 minutes. During that time, the centrifuge stopped spinning, so let me grab the tubes out of the centrifuge. Alright. I'm going to show you what these look like. So, remember the volume was up so we had the solution up at the top. It's gone through, so our DNA is going to be stuck on there. We don't need that solution. So all you do is you just dump it out. Put the column back in. And now we're just going to go through a series of washes on the DNA. Clean it up. Get rid of any of that salt we don't need. And the first one, so we got to set this 500 microliters. Yep. On the column for each of them. Get this in the centrifuge, and it'll be perfect timing for moving our transformation forward as well. So let's stick this in the centrifuge. The timer's going to go off before then, but that's okay. Not a problem. All right. Okay. So, the last part, I'm just going to grab another, since we need this. I'm going to take this guy off the ice. We don't need the ice anymore. So to make these happy, these cells nice and happy again, microliters, we're going to give them, like, the ultimate food, essentially. And it's called SOC media. And it doesn't have any of the antibiotics I was talking about. It's just regular media. But it has, like, extra yummy stuff for the E. coli to make them extremely happy. And can basically forgive us for what we just did to them. Which is we heated them up, which they don't like. And then I'm going to go put this in our shaking incubator for an hour. And we use tape just because we put it on the bottom. It works. It works. So I will be right back. I'm going to go get this started. Alright, so they're in our shaking incubator, 37 degrees Celsius, their favorite temperature. They're going to grow up for an hour. Our centrifuge stopped. So let's grab our tubes. There we go. Start our timer for an hour, and I'm going to change this to 750. 750 microliters. There. First, get rid of the cleaning solution, cleaning buffer. We don't need that. Jump it in the waste beaker. Go. Okay. There we go. All right. Now for the other cleaning solution. Cleaning buffer. And this is going to be our last wash, the last time we add a buffer to wash it. We don't need to do another one after this. It should be nice and clean. Got rid of all that extra salt. Perfect. So this buffer contains ethanol. And it's great for cleaning up your DNA. Not so great for downstream stuff that you're doing with the DNA. So after I do this spin, which I'm doing right now, for a minute, I'm going to do an additional spin to get rid of any of this residual, ethanol, because we don't want any to go through with our DNA, because it will definitely mess up proteins or anything that we're going to be doing downstream. So basically, I'll just dump out the ethanol solution, spin it again, get rid of the rest of it, and we should be good. I'm just going to put these back. We're all done with them. And also dump out the ice. We do not need ice anymore. We're all set. There we go. So that is done. Got buffer on here again. So messy. So messy. That one just doesn't stick, but it can leave white residue because of the salt in it. There we go. So that's all set. Actually don't need this pipette anymore. We are all good on that. Going to need this one, and I'm going to set this to 25 microliters. So once the DNA is all washed, you might be wondering, well, how do I actually get the DNA afterwards? So In order to elute the DNA, or get it off of the membrane, we just add water to the membrane. Let it sit for a minute, and then we spin it down, and the DNA is there. Pretty easy. Alright, so let's dump off this ethanol buffer. But you can see, like, there's still some ethanol in there. We don't want that moving forward. And there's some on the sides, so basically we just want to make sure that we get any of the ethanol buffer that could be stuck in this tube to be gone, because we don't want it sticking to the column. Alright. Back in the centrifuge for another minute. Alright, that's going. We don't need this. We're actually all done with that, which is nice. And we just let that spin for a minute, and we will be all set and ready to elute our DNA off of the column. Yay! So that'll be great, and we have over 55 minutes for the transformation in that case. So if you guys have any questions during that time, feel free to put them in chat. Not a problem. I can also go over to a review of what I did earlier if you would like, if you missed it at the beginning. I don't mind doing that at all. No, not at all. Or depending on the time. Actually, yeah, it's almost noon for me. Lunchtime. We'll have to see. We'll have to see. I might stay on a little bit longer, but I would like to show you the plating. We'll have to see, though. There's some other things. I can also just leave it on, and we can listen to the music while I go do a couple of administrative things. That's fine. And then I can come back when the plating is ready for our transformation. I don't mind doing that either. And just keep my eye on chat. Alright, so this is done spinning. Should have gotten any of that residual ethanol buffer off of the column. We shouldn't have that carry forward with our elution steps. So what I do next is we move the blue column to a new tube. Get rid of that. So this is why we don't want any ethanol attached to this, because this, we're going to take the DNA off and it's going to go into the tube. So that's why we don't want any ethanol. Alright. Great. Okay, so now I'm going to add water. If I'm microliters, I'm going to add it right down onto the middle of the membrane. Basically like that. You know what? Because I'm showing it to you. Just going to do that. Usually I don't touch the end, but I'm trying to show it to you guys, so I'm actually touching it, changing my tip just in case. I don't want to cross-contaminate my plasmids. That would be bad, because then I don't know which one's which. Ooh, don't want that. That's why I go, no! I have to do it all over again. Because I do have some more of them in the fridge. That's why I didn't spin down all of it. Alright. Now we let it sit for a minute. And then we'll centrifuge it. And then we'll be done. And then we'll have our DNA nice and clean, ready to go forward with. So the next step that I would do would be a restriction digest. So that basically is using enzymes to cut the DNA at certain places and run it out on a gel to see how the DNA bands are and compare it to what I expect it to be, if it is the correct plasmid, if what we did was correct. I don't have time to do that today. If you would like to see past streams about that, I do have them up on my YouTube channel. and we go through restriction digest, and then on the sequencing VOD, I actually go a little more in depth into how you look at the gel and analyze the gel. So if you wanted to see the more in-depth version of that with the actual better picture up on screen, it's in the sequencing VOD on YouTube. All right. I'm just gonna set that back to our transformation. And let's get this centrifuged. Once again, just a minute at that 18,000 times G. And then we'll be all set. But yeah, so I'll probably check these tomorrow. I just won't have time to stream. So that's why I won't be able to show you what it looks like. I can always let you know. If you're curious as to how well it goes, I can let you know. Because I will be very excited if one of these ends up being the plasmid that I need. Oh, yes! Perfect. I'll be so happy. I'll be so happy. Ooh, which reminds me. So one of the things, as I was talking about plating, which will happen in 50 minutes, I need to get the plate out and put that at 37 so it's at the temperature that the E. coli likes. So let me get that plate out and I can show you what it looks like without anything on it, basically. Let's see. Grab a plate. And we make these plates here as well. They're pretty easy to make. So I made these yesterday. So this is Petri dish with agar. So this is agar right here. Basically has all the solutions that E. coli needs just in solid form. So you see what I was talking about earlier, where this is liquid media right here. This is solid media. And it does contain the ampicillin antibiotic, so the antibiotic is already on here. If the E. coli did take up the plasmid that we made, then it will grow on here. If it doesn't, then it won't grow. But I need to put this at 37 and get that all nice and warm. All right, so I put that at 37, we're all good. I'm going to grab our tubes out of the centrifuge. Get that all set. All right, there we go. Okay, so don't need the columns anymore. There we go, there's our DNA. And the colorless liquid at the bottom of the tube. So our DNA is all set. It should be in here, if it ever existed within those E. coli cells, which hopefully it does. All right, so won't be able to check these today, but I'm going to put them in our freezer so I can check them tomorrow. And then we're all done. So that was plasmid DNA extraction. All done, set and ready. So the second one that I've done on stream. But we're all good. And in the freezer it goes. Ready to be worked on tomorrow. Cool. There we go. Sorry, my headphones keep on coming down. Gotta try to fix that. All right, so I think we'll take a short break. I need to get something to drink. And I can come back and see if there's any questions in chat. If not, then we can just hang out until timer goes off and I will show you how I plate the transformation. So let's do a short break and I'll be right back. Alright, I'm back. I got something to drink. Needed that brief little pause there. Hopefully you guys also took a nice little break, got something to drink. So yes, if anybody has any questions, we can go over what I did earlier in streams, not a problem. Or any questions about what I just did on stream, not a problem as well. Or just talk some science, we can do that too. Not a problem. Welcome to new people who are watching the stream. Welcome to the bench. I'm Neo Q-Star or Dr. Jess. It's all good. Either one of them works for me. And I just stream what I do at the bench as far as experiments. Today we're doing a bunch of plasmid stuff. Earlier we made some plasmid, we did some plasmid construction. We're in the middle of doing a transformation, so we took the plasmids that we constructed and are transforming them into E. coli. And we just finished up doing a plasmid DNA extraction. So yes, that's what we're doing today, just a bunch of plasmid work in this case. So yes, if there's any questions, we can definitely go over it again. Once again, I'm very glad that you could join me today at the bench. I know it's been a while since my last stream, but I wanted to do a stream with you guys today. Get some science in, spend some time with you. Go over a new protocol that I haven't done on stream yet as far as plasmid construction. Share that with you. And just, you know, hang out at the bench. It's all good. Ooh, you know what? Since I cleaned off my bench, we can bring our mascots back. Ooh. Bert and Ernie and Eli Ecoli can come back to join us since we're using both of them today. There we go. There. There's our favorite channel mascots. We can hang out. Would it be weird if if nobody has any questions, if chat is no chat, then that's fine. I do not mind at all. But would you guys find it weird if I just left us here and we listened to music and these guys kind of just stared at you? Would that be too creepy? Would that be too creepy? Hopefully not. Hopefully it won't be too creepy. Because we can definitely do that. Because basically we were just hanging out for another 40-something minutes until I show you how I plate the transformation. And that's basically going to be the end of stream at that point. But, man, we can sit here and just chill. I'm okay with that too. With Bert and Ernie and Eli Ecoli. Alright, well, I don't mind it being quiet in chat, that's fine. We can sit here and enjoy the music, and I think I'm just going to leave our two mascots here with our timer so you have an idea of when we're going to be plating up our transformation. If you have any questions, please feel free to put them in chat. I will be here monitoring chat. Not a problem, I don't mind answering them. But yeah, so next actual bench science will happen once the timer hits 0. And we'll be all set. So yeah, we can sit here and chill. Enjoy the music, everyone. Hi everyone, I just wanted to say that I saw there's a couple new people in chat. If you have any questions about the science that was done at the bench today, feel free to put them in the chat and I will answer them. We're just hanging out right now until timer reaches 0 and we will finish up the current protocol we're on, which is a transformation of the plasmid, one of the plasmids we constructed today, and we transformed it into Eli Ecoli. So yeah, if you have any questions, feel free to put them in chat. I'm just hanging out over here. We're listening to the music while we're waiting. But I am here, ready to field any questions. So you can just chill out with us as well. And welcome to the bench. Alright, so I've got about 15 minutes, and then we're going to be able to plate up our transformation. I think there's a couple new people in chat. Just wanted to let you know that we're just hanging out right now, finishing up what we were doing earlier. If you have any questions about what might happened at the bench today as far as science goes, feel free to put them in chat. Not a problem. I just changed this. Let's see if it works. Nice. All right. So that just gives you a rundown of what we did today at the bench. But yeah, we're just kind of chilling, hang out until this is done and ready. I'm watching just in case there are questions that come through. But yeah, I'm also enjoying this little chill time. Every once in a while, it is nice to just chill and listen to the music while waiting for something. Gives my voice a chance to recover as well, since getting over this cold, every once in a while my voice decides to start to go. So it's all good to have a nice little chill session every once in a while. But yes, thank you for joining me at the bench today. We're going to be wrapping this up with the plating of the transformation. And if I don't get any questions during that time, then I'll probably end stream. And if you missed the beginning of the stream, don't worry. The VOD will be up on my YouTube channel later. I'm just gonna, once more, put that up there. And also, of course, Twitch will have this up for 7 days, I believe, on my channel. So feel free to go back and look at it if you would like. Or you can look at them on the YouTube channel. I will have it up as soon as it's basically done and rendered on YouTube today. So yeah. Any questions, put it on there. And of course I need to excuse me while I fix closed caption. Alright, hopefully that fixed it. Sorry, every once in a while I have to clear the transcript because it gets a little too long with all the talking, and I'm not entirely sure what happened with the music there either. I apologize for that. Looks like it's back. Awesome. And of course now it's cut off the top. How about that? Is that better? Ah, much better, much better. Just do a little bit of tweaking as that goes on. Alright. Any questions as we keep going? Just want to keep checking every once in a while. I am here. I am here. We can chill and listen to this music. I'm quite enjoying this. So yeah. Enjoy the music, enjoy the vibes. Alright, well, looks like we're coming close to the end. We've got about 2.5 minutes left. So, I'm going to get ready for plating our transformation. We will be all set and ready to go. I'm going to go get the plates that I had heating up. So that should be a nice cozy 37 degrees Celsius for our E. coli once they are out of the shaking incubator. So I'll be right back with that. Alright. Oh. Alright, we gotta say goodbye to our mascots. Okay, here's our plate. It is nice and toasty. Well, it's at 37 degrees. I'm going to label it so that I know what this is. Yes, okay. Sorry, I had to look back at my notes as to which one we transformed. Oh. Today's date. So it'll be all set. Flip that over. So I actually labeled it on the part where the bottom of the plate, right here, because on the top is usually where you would see and look if there's any colonies. And if there's writing here, it's kind of hard to see when you do it. So label it down here. So all set. Okay, so that's done. I'm ready to go. Put that away. And what I'll need these. Microcentrifuge tube and our P1000. I'm going to set it to about 300. Oh, I'll set it. Put it there because I want to plate the entire transformation that we have. All right, and then be all set and ready to go. Let me go grab it from our shaking incubator. So it's been an hour at 37 degrees Celsius in our shaking incubator. So our E. coli should be happy after we did the heat shock and growing nicely since we gave them their nice little spa treatment, their favorite food. But now we're going to be mean to them and put them on the antibiotic. Going to get fire transformation and plate it out. There we go. And then I just put it like that. Move this stuff out of the way. And then spread it around using this tube. So once again, I'm showing you what I do in the lab. There we go, so it's all plated. So you don't have to use a microcentrifuge tube. That's just something I do, and I did as a grad student. There are like disposable spreaders that you can actually get for this type of thing. Microcentrifuge tube is ready for me right there. I'm going to go put the plate in our regular incubator, so not a shaking incubator. We don't want to shake the plate. That's set at 37 degrees Celsius, and these guys are going to grow up overnight. And hopefully there will be colonies. So I'm going to go do that real quick. Alright, well that is the end right there. I'm going to get rid of my gloves. I don't need them anymore. We're all set. So that was the last part of the procedure. Protocol, procedure, method. All the same thing. Wash my hands. Alright, so that is all done. growing up. I won't be able to show you what the plate looks like tomorrow since I won't be streaming. But if you want to know what a plate looks like after a transformation, you can go back into my VODs, and there is one for picking colonies. And that's where you'll be able to see what the plate looks like after it grows up overnight, if there's colonies there, and what I'm going to do afterwards. So just a brief recap of what we did today, which was basically just a bunch of plasmid work. We started off by doing plasmid construction. That was using 2 different protocols. One was just the regular ligation, which I've done before, and then the other one is Gibson/Hi-Fi, which uses 3 different proteins, and the homology, so the 2 spots in the sequence that you add with a PCR primer. So all these jargon is at the beginning. I apologize. If you have any questions, please put them in chat of what I'm talking about. And that puts them together with our vector and our gene. And then we transformed that one into E. coli today, and we just finished that up. The regular ligation is at 16 degrees overnight, just because it's a much harder ligation, and I'm really hoping that it works. We shall see. Then, while we were waiting for that, we did a plasmid DNA extraction of some colonies that I had picked a long time ago. Well, not I had picked, a grad student in the lab had picked, and hopefully we will get some positive plasmids out of that. I'm going to check those tomorrow, but like I said, I won't be able to stream that. If you want to see how you check a plasmid after doing extraction, I have a VOD on my YouTube channel for that as well. And then, as I mentioned before, if you want to see more in-depth analysis of the gel that you run following that, it's part of the sequencing VOD, so the enrichment one, and you can see it there. But yeah, so that's what we did today. If there aren't any questions, I think I'm going to wrap it up here. So thank you so much for joining me today at the bench. I know the last 40 minutes were pretty much just chill listening to the music, but hey, it's all good. It's great for me as well. But I'm glad I got to share some science with you. I know it's been a while since I did a stream. Hopefully it won't be such a long time for the next one. We're moving into more sequencing and consistency, and also a fix to the scratchy audio. I'll be really excited about that. But yeah, so if you want to see this VOD and catch the beginning of it, it'll be up on Twitch, and then it'll be up on my YouTube channel later. But yes, I'll just give it about a minute just to make sure there aren't any questions in chat. All right. Well, thank you again for joining me. Have a good rest of your day, evening, morning, whatever time it is for you at this time. And I will see you again next time. Bye.